Lesson 6, Nutraceuticals to Power Brain Function. The importance of expanding your knowledge base on nutraceuticals which serve to support optimal cellular functioning cannot be underscored here, as this is how to ensure that your clients have the foundational building blocks which will allow the body to continually produce neurotransmitters and cofactors at levels essential to supporting optimal brain health. Aging occurs when cellular death from reactive oxygen species occurs at a faster rate than cellular growth. The brain is the most metabolically active organ in the body, causing it to generate a large amount of reactive oxygen species in the process. It is also the most vulnerable to oxidative stress. These reactive oxygen species often get neutralized by the brain's natural antioxidant systems. In a healthy individual, these systems operate efficiently. But in an aging individual, the reactive oxygen species are often in excess of the antioxidant capacity, resulting in cellular aging. The solution is to incorporate nutritional solutions to support the brain's antioxidant capacity. Here, we're going to discuss the nutraceuticals important in supporting brain function through enhancing antioxidant systems and neurotransmitter production, along with the stabilization of cell membranes and blood glucose levels. In this lesson, I'm going to review 7 powerhouse supplements which have a strong evidence base in the peer-reviewed literature. These include Phosphatidylserine, which provides cell membrane stability, Acetyl-l-carnitine, which supports cellular respiration, N-Acetyl Cysteine, a potent antioxidant, alpha-lipoic acid, which serves to maintain healthy blood sugar levels, Ginkgo biloba and Vinpocetine, which enhance blood flow throughout the brain and body, and Huperzine A, which serves to increase the neurotransmitter acetylcholine, which supports cognition. We use these supplements in combination in an open-label clinical trial in professional NFL players and demonstrated that they have significant impacts on cerebral blood flow and multiple domains of cognitive proficiency, which I will discuss at the end of the lesson. Phosphatidylserine is a fat-soluble phospholipid that's an essential component of cellular membranes. It's needed by every cell in the body and is the most abundant phospholipid in the brain. It's important in the neuronal membrane function, including maintaining the cellular environment, signal transduction, vesicle release, cell-cell communication, and cell growth regulation. With the aging process, the neuronal membrane changes in composition and begins to lose its receptors. This results in compromised neuronal communication, which interferes with storing memories, resulting in age-related memory decline. Phosphatidylserine has been demonstrated to repair cell membranes, which helps prevent cognitive decline in those with memory impairment. It's thought that exogenous administration of phosphatidylserine may normalize brain lipid content and return neuronal function to normal. Phosphatidylserine has been shown to increase acetylcholine, norepinephrine, serotonin, and dopamine in human and animal models with Alzheimer's disease. Phosphatidylserine minimizes age-related neuronal dendrite loss and the atrophy of cholinergic neurons. Phosphatidylserine has been found to be important for energetics, as the mitochondria requires it, and phosphatidylserine bonds with the omega-3 DHA, functioning to strengthen synapses. The body can synthesize phosphatidylserine, but it is mostly obtained through the diet in fish, soy, cabbage, and white beans. Acetyl-l-carnitine is a natural compound essential for normal mitochondrial function. It facilitates the transport of long-chain fatty acids across the mitochondrial membrane, promoting enhanced cellular energy production. This powerful dietary antioxidant functions to improve free radical scavenging, as well as the inhibition of pro-inflammatory pathways, which maintains and repairs neuronal damage. It is also thought to influence the cholinergic system by promoting the synthesis and release of acetylcholine, which is required for memory storage and retrieval. It can also increase protein kinase C activity, as well as prevent the number of NMDA receptors that are lost due to aging. Acetyl-l-carnitine has been most studied for its anti-aging properties, as it provides brain and nerve support. research suggests that it may slow the decline in cognition, mood, and daily function that occurs during aging and degenerative disorders. Acetyl-l-carnitine is thought to prevent neuronal degeneration and help in the repair and regeneration of neurons, therefore decreasing excessive excitability and firing. Acetyl-l-carnitine occurs naturally in the body and can be synthesized from the amino acids lysine and methionine, but can also be obtained in the diet from consumption of red meat and dairy. N-Acetyl Cysteine is an amino acid that is needed to produce glutathione, a very powerful antioxidant. N-Acetyl Cysteine freely crosses cell membranes and functions to chelate heavy metals and environmental pollutants, which means it binds to and removes dangerous toxic elements within the cells, making it a molecule critical to brain health. In addition, N-Acetyl Cysteine is a vasodilator, working to relax blood vessels and allow more oxygen delivery into the body. It also functions to enhance the immune system. N-Acetyl Cysteine's primary use in brain health is for foundational support in individuals with Alzheimer's disease, those working to reduce homocysteine levels, and detoxifying heavy metals, including mercury, lead, and cadmium. It also protects against environmental pollutants, including carbon monoxide. It is most useful when glutathione levels are low, as in conditions of oxidative stress. Ginkgo biloba is an extract from the Ginkgo biloba tree, native to China, which is most recognized for its neuroprotective antioxidant properties and ability to enhance circulation. Ginkgo extracts contain several active constituents, including flavonoids, terpenoids, and organic acids. These compounds have been shown to act as free radical scavengers, which give Ginkgo biloba its antioxidant and neuroprotective properties. Promising research studies report that supplementation with Ginkgo biloba protects cell membranes from lipid peroxidation and neurons from oxidative damage. Ginkgo biloba's ability to improve cerebral and peripheral circulation through decreasing blood viscosity and affecting vascular smooth muscle contraction may result in the benefits associated with blood flow. Ginkgo has also been shown to inhibit cellular toxicity induced by beta-amyloid proteins, which are implicated in Alzheimer's disease, and has been demonstrated to enhance cognition in healthy individuals as well as those with age-related cognitive impairment. Furthermore, Ginkgo biloba has been shown to improve working memory independently and when combined with Vinpocetine. Vinpocetine is an alkaloid compound extracted from the periwinkle plant and functions as a phosphodiesterase inhibitor. It is an effective vasodilator and has been used in the prevention of cerebrovascular diseases, cognitive enhancement, and has also been shown to have anti-inflammatory and neuroprotective properties. Positron emission tomography studies demonstrate Vinpocetine's global uptake across the brain and its ability to effectively regulate blood flow and metabolism. So Vinpocetine appears to have many varied pharmacological effects. For example, some studies indicate that Vinpocetine might enhance cerebral blood flow without affecting peripheral blood flow. Preliminary evidence indicates that Vinpocetine stimulates cerebral metabolism and increases glucose and oxygen consumption by the brain. Potential mechanisms for these effects of Vinpocetine include direct or indirect cholinergic activity, augmented norepinephrine effects on cortical cyclic AMP, and increased turnover of the brain's catecholamines. It might also improve microcirculation in the brain and increase cerebral blood flow by improving red blood cell deformability, reducing cerebrovascular resistance, and inhibiting platelet aggregation. Alpha-lipoic acid is a powerful antioxidant, which neutralizes free radicals, which ultimately serve to detoxify and protect the body. It serves to recycle both vitamin C and E, as well as supporting glutathione production, making it a powerhouse in maintaining optimal antioxidant production in the body. Given that alpha-lipoic acid is soluble both in water and fat, it can move into all parts of the cells to remove damaging free radicals. Free radicals are atoms or groups of atoms that contain an unpaired electron. While lasting less than a second, if in excess, they can cause irreversible damage to the heart, nerve, and immune cells of the body. Free radicals can be produced by radiation, X-rays, environmental pollutants like tobacco smoke, and poor diet, such as cooking fats at high temperatures. Excessive free radical production can result in damage to genes, which then produce mutated proteins. They can also destroy cellular membranes. Ultimately, excess free radical production is what leads to cellular aging. So alpha-lipoic acid is most powerful in controlling blood sugar levels in individuals with diabetes. It can also detoxify heavy metals and protect nerve tissue against oxidative stress. Alpha-lipoic acid is synthesized by the body, but is also found in the diet in red meat, organ meats, spinach, broccoli, potatoes, yams, carrots, beets, and yeast. Huperzine A is an alkaloid extracted from the Chinese club moss, as seen in the image here. It functions as a reversible, selective inhibitor of acetylcholinesterase, but has also been shown to protect against oxidative injury, neuronal apoptosis, and glutamate-induced toxicity. It elevates acetylcholine levels throughout the brain, with maximal values demonstrated in the frontal and parietal cortex. It also protects against glutamate excitotoxicity by blocking glutamate-induced neuronal calcium influx and cellular death. In essence, Huperzine A boosts acetylcholine levels, which can serve to support cognitive function. So now I want to take all of the individual supplements we've discussed during this lesson and demonstrate their efficacy in impacting brain function in specific and measurable ways. During my tenure as Director of Research for the Amen Clinics, we initiated a study in 2009 measuring the effects of repetitive subconcussive impacts in living professional football players on brain function. Given the significant damage to the brain observed on both imaging and assessments of cognitive and psychiatric function, we added a rehabilitation component to this work, evaluating the effectiveness of the brain-directed nutrients I discussed in this lesson, which also included a high-potency multivitamin and omega-3 fatty acid supplementation. In addition, lifestyle interventions, including nutritional coaching, access to a weight management program that I ran, and a sleep apnea testing were offered to the participants for the length of the trial. We performed an open-label clinical trial in a group of 30 of the active and retired players with traumatic brain injury. Following an average of 6 months on the program, the athletes demonstrated significant increased blood flow as measured by SPECT imaging in areas important in focus and impulse control, emotional regulation and empathy, memory, visual processing, spatial processing, and reaction time. These areas are highlighted in yellow and orange in the image above. 25 out of 30 participants reported improvements in memory, attention, mood, motivation, and sleep. We published these findings in the Journal of Psychoactive Drugs in 2011 in a manuscript entitled Reversing Brain Damage in Former NFL Players: Implications for Traumatic Brain Injury and Substance Abuse Rehabilitation.